once you've picked a general topic, you need to focus that question down. And what that entails, again, we're back to the hard work piece. You need to evaluate the medical literature and other sources to evaluate the current knowledge in that research area. Maybe the question you're interested in has already been answered by somebody else. In many areas, though, much of what we think we know is less than what is generally believed. And one of the places to start with this is review articles or treatment guidelines. But really what these do is represent a synthesis of others' view on the data. And much in treatment guidelines is graded according to the strength of the evidence. There was a recent study done about 4 years ago which actually looked at the recommendations in both cardiology and infectious disease treatment guidelines. Only 11% of what was in them had been generated from randomized controlled trials, and almost half of the recommendations were based on clinician opinion. So that's a bunch of nice guys sitting around a table saying, I think thou should do this. But that's not evidence, and that's not science. So many times what you can actually get out of guidelines is actually some of the questions that still need to be answered, because even though they're in the treatment guideline based on best opinion, they haven't really been answered in a scientifically valid way. So John Unitas actually brought this up in an essay that he wrote in 2005, saying that for many current scientific fields, claimed research findings may often be simply accurate measures of prevailing biases. And so what he meant by this, too, is that if you don't design a study properly, you may get an answer with a p-value around it, but a p-value doesn't tell you anything about clinical meaning, nor is it a measure of bias. So a poorly designed study can get you an answer with a p-value of 0.001, but all you've done is measure what that bias was. So you got an answer that was accurate, but just more accurately wrong. And that's the point that he makes in this particular essay. So when you're looking through the medical literature, you're looking for what's already known. And I jokingly call this the knowledge gap, right? You're looking for things that we don't know already that would make good research questions for you. So the first is, how do you define the problem that you're interested in looking at? What are its clinical features? What signs, symptoms, laboratory values go along with that clinical entity? Is it known how to diagnose the problem? And what is the impact of diagnosis on outcome? So for instance, if you come up with a great diagnostic test to detect something, but that something doesn't mean anything to the patient because they're going to die of something else when they're 95 years old, it's not of much utility. So for instance, the debate about the use of prostate-specific antigen testing, where a large study done here through NIH showed that using PSA to screen men for prostate cancer really didn't change outcomes for them, because many times you find prostate cancer that's in its early stages that's not going to harm the person at all, and they're going to die somewhere down the line of something else. In fact, I told my doctor after that study came out, if he puts PSA on the lab test, I am crossing it off. Because a friend of mine actually jokingly called the PSA test should be renamed prostate-specific anxiety instead of the prostate-specific antigen, because you tell somebody they got a positive test, they're going to worry about it, even if the test doesn't mean a whole lot to them in the long run. So the next thing is, how big is the problem? Is this a really common problem so that the number of people affected is very high and would have a bigger public health impact, or is it something that's rare, but even if it's rare, has a huge impact on the people that actually have it? And that's the fourth question, is looking at the impact in terms of attributable morbidity and mortality. And this is often assumed in terms of people will say, well, disease X is very prevalent. But if the more minor stages of that disease are prevalent, then it's not as big an issue. In fact, I wrote a paper back in 2008 where we looked at what the impact was on antibiotics and pneumonia back in the 1930s and 40s when penicillin and sulfa drugs were first introduced. It showed that penicillin reduced mortality in young, healthy people, but only by 5%. If you were older and sicker, it reduced mortality by 40%. That's huge. I mean, a 40% absolute reduction in mortality is gigantic. But what was really interesting was to show that 95% of young, healthy people with pneumonia in the pre-antibiotic era got better on nothing. So the idea is, it depends who you are in terms of what the impact of the problem is. And that also gets to the issue of what are the risk factors for getting the problem in the first place. And risk factors are not necessarily causal. So for instance, you could say older people are more likely to get pneumonia. Older people are more likely to have gray hair. Gray hair does not cause you to get pneumonia, but gray hair could be a marker for getting pneumonia. So risk factors don't necessarily need to be causal to begin with. The next question you could ask is, what's the prognosis in terms of morbidity and mortality, and what factors might modify that prognosis? Because if you are doing a clinical study to describe just the natural history of the illness, those things may be impacted by who you actually study. And when you are studying treatments, those things are referred to as confounders. In other words, those things may impact outcome independent of the treatment that you're actually trying to evaluate. What interventions can actually mitigate the problem? And this is much of the research that gets done, usually, but not always, in randomized controlled trials, evaluating interventions like drugs, devices, biologics, and also even different behaviors like hand washing. Does hand washing decrease infection rates in the hospital? And then finally, there's the two questions that are, when there are effective treatments already available, there are still questions to answer about how you use them properly, how they compare to each other, and do certain interventions work better in some people than others? So that's the issue of effect modification. So for instance, we know that beta blockers do not work as well to control blood pressure in African Americans as they do in Caucasians. And then there's a big push now to do what's called comparative effectiveness research. So for instance, in cardiology, they look at more expensive medications compared to older things like diuretics and show that the newer drugs may be more expensive, but they really don't help you lower blood pressure or decrease death, strokes, and heart attacks any more than the older drugs actually do. So one of the things that you run up against when you're evaluating the medical literature in the field that you're interested in is that people believe a lot of things that may not be supported by evidence. When I was a fellow, I actually went back and there was a statement that any person with who had a kidney transplant, if they had fungus in their urine, had to be treated because they would get a fungus ball in their urine that would knock out their new kidney. So I found that in a textbook. I looked at the reference. It referenced another textbook, which referenced a paper, which was an opinion piece, which didn't have a reference. So do people really get fungus balls and knock out their kidney when they have a kidney transplant? I don't know, because when I tried to trace that information, it turned out to be one person's opinion in an editorial and not anything based on even a natural history study of people that had kidney transplants. So Daniel Boorstin, who was the head of the National Library of Congress for a number of years, wrote, "The greatest obstacle to discovery is not ignorance, it's the illusion of knowledge." And that also, for you when you're doing your research, is an issue because you have to convince people that maybe what they know isn't what they really think they know in terms of why you want to answer the question that you do. So that gets to the issue of what do we know about previous questions. Oftentimes what happens is you get into this issue of, I took a logic course when I was in college, and one of the logical fallacies is called argumentum ad verecundium, and that's arguments from authority. Well, I'm a big wig in this field and I say so, that makes it true. That's not science, though, but we often run up against this, and in fact, television is full of this stuff, right? All the talking heads on TV who say it's got to be true because I say it's true. But the real question for you as a scientist is, what is the quality of the data? What's the validity, reliability, and precision? And are there biases in the previous data? So one of the good things about the rise of meta-analyses since the 1970s is this issue that the quality of the data and potential biases in the data is taken into account when people try to look at that data synthesis for a meta-analysis. Has the evidence been independently confirmed? So is there one study that shows this treatment is effective and 10 studies that show it's not? And that's one of the basic principles of the scientific method is make an observation, develop a hypothesis, test the hypothesis in experiment, and what's point number 4? Confirmation. Because you might just get lucky or get wrong in a single study, and one of the basic tenets of when I was a fellow, we actually wrote things down in lab books. We don't do that anymore. Everything's on a computer. But the reason we wrote our experiments down is so somebody else could replicate that same experiment, not just me. So the other question is the evidence consistent across different populations? Like I said, effect modification means it might be effective in one group and not in another, or the side effects might be different in one group than another. And for interventions, we might want to look at different doses, different durations of therapy, or combinations of therapy. So after you get through that, and that's a big process, going through all that medical literature, you need to focus on one part of that knowledge or research gap, or one piece of the puzzle. You also need to think about not just your study, but how is your study going to fit into the bigger research picture? So the best kind of studies, the joke always goes, ask more questions than they answer. Meaning, once you answer one piece of this puzzle, it leads to asking all these other things that may relate to it. So for instance, you do a research study and you show that drug X is effective in treating disease Y, but then people might want to say, well, yeah, but does it work at a lower dose? Does it work better at a higher dose? Does it work better in combination with something else? Those are all questions that might come up later. So you need to actually think about, how does this fit into the overall research plan? The best kind of questions are the ones where even a so-called negative study still gives you useful results. So for instance, the studies in using prostate-specific antigen as a screening tool, even though those studies didn't show that that was a useful screening tool, it still came up with very useful information about who may or may not need to be tested with that test. The issue here is a single study cannot possibly answer all the questions about a topic, and in fact, one of the major flaws in research is people try to answer too many things in a single study. It's kind of like trying to put 25 anchors on a boat. You're going to sink the boat if you try to answer too many things at one time. So you really want to see, how does your research fit into the overall model or theory of the problem? Once you then get to this issue of feasibility, you have to address what kinds of information are you going to need to be able to do your study. And that relates to what kinds of people are you going to study, what populations, and if it's a comparative study, you need to have a test group and a control group to compare to each other. What exposures are you going to analyze? In other words, are you testing people if they get this drug versus that drug? Or is the exposure something like people who smoke versus don't smoke, or some environmental exposure? And what outcomes are you going to measure? Is it going to be death? Is it going to be some measure of disability, etc.? What kinds of information are available to you? Do you work in a hospital that's got enough information, or a system that's got enough information to do this? We recently tried to do a study here where we looked at all the positive blood cultures at the NIH Clinical Center to look at the impact of antibiotic resistance on outcomes. It turns out that the Clinical Center is actually a fairly small hospital. So doing this only here did not give us enough information to be able to answer that question. So now we're trying to collaborate with other hospitals to try to pool our data with theirs to be able to address that question. So what resources do you need to obtain the data? Is this stuff that's already available in medical charts, or are you going to need to go out and get that information? And even if the information exists, do you have access to it, or can you get access to it? And again, just to reinforce, feasibility doesn't mean using invalid methods because, well, that's all I can get around to doing.